Good day once again, my students. I am Comrade Isaac Igbawua from the Department of Microbiology, going to be taking you on General Microbiology 2, 3 credit load course, and I believe together we should be able to understand what the whole course is all about, and in terms of content we will be considering these topics: transfer and change of genetic information in bacteria. Under that we'll be looking at transformation, transduction, and conjugation. We also have a look at the role of plasmids organisms generally and human diseases. In the course of the course, we also have to look at some common infectious diseases. We'll also be looking at elements of immunology. The course will take us to look at microbiology of soil and water. We'll be considering industrial fermentation in the course of the course. And finally, we'll look at food and dietary products. For the purpose of this week's course topic, we will consider transfer of genetic information in bacteria, and that will take us through the 3 modes of genetic transfer, which has to do with transformation, transduction, and conjugation. By way of introduction, bacteria reproduce by splitting into 2 via a process called binary fission. Binary fission makes clones or genetically identical copies of the parent bacterium. Since the child bacteria, that is the one that is produced, is genetically identical to the parent, binary fission does not produce an opportunity for genetic recombination or genetic diversity, aside from the occasional random mutation that may be observed. Bacteria divide very rapidly. The doubling time is also called the generation time, and this could be as fast and as low as within 20 minutes. And so you can now wonder how many bacteria cells that could be produced at each turn in after a generation time. Bacteria, in considering modes of genetic transfer, reproduce by asexual reproduction, such that they do not exhibit true sexual reproduction, but can be very active in the exchange of genetic information. This is because they do not produce diploid phase and therefore no meiosis. Remember the meiosis and mitosis in your elementary biology. Now, 3 modes of genetic transfer between bacterial cells are recognized, namely transformation, transduction, and conjugation. We will briefly consider them one after the other. Transformation, number 1. It is a process of introduction of derived DNA fragments from a donor bacterium into a recipient bacterium. In other words, transformation involves the introduction of DNA component or parts from a donor bacterium into another bacterium that hitherto did not have such parts or genetic material. It is a phenomenon by which DNA isolated from one type of cell, when introduced into another type, is able to bestow or let onto it some of its properties into the latter, which is the receiving bacterium. However, bacteria exchange genetic material between two cells, but only one partner receives the new information. Get this clear. The information being given was originally in the donor. So the child bacterium is the one in this type of arrangement, is the one that is going to receive the new information. The other one only have what is given. It doesn't take from the other one. So the donor gives that new thing only to one partner, the receiving bacterium. The DNA taken can be incorporated into the recipient's chromosome by a process called recombination. When the DNA fragment is now let into the recipient bacterium, there is recombination with the chromosomal DNA of the receiving bacterium, such that the cell is now transformed into a different cell, having components or structures that ab initio were not there. It was first demonstrated by Frederick Griffith in London in the year 1928. I'm talking of transformation. It is a natural process, but can also be induced in the laboratory. One thing that is clear and paramount is that only competent cells undergo transformation. In other words, there are some cells, even though the DNA fragment will be there, they may not be able to receive and get recombined with those or with that DNA fragment or component. So it's only cells that are competent, that are able to receive and incorporate such DNA component that can undergo this process we call transformation. If they are not able to recombine, then the process could not take place. And only certain bacteria are able to do such. For example, pathogenic bacteria such as Haemophilus species, Streptococcus species, and Neisseria species are examples of competent bacteria cells that exchange genetic material through the process or mode of transformation. The importance of this transformation. Imagine a harmless bacterium taking up DNA from of a toxic gene from a pathogenic species of bacterium. If the receiving cell incorporates this new DNA, which is this toxin gene, into its own chromosome, which can happen in a process we call homologous recombination, it too, that is the receiving bacterium, may also become pathogenic. And so that is the importance of this thing. So that bacteria that originally was, or naturally, not having any disease-causing ability because of transformation, because of this transfer of this toxic gene will be made to become toxic by transformation or transfer of the toxic material or DNA component or fragment from the pathogenic bacterium into the harmless bacterium, which upon incorporation may also become pathogenic. If you look at the illustration on the structure, on the outside is a plasmid. Plasmid generally are extra chromosomal DNA molecules that are capable of self-replication outside of the host or chromosomal DNA. They are self-sustaining. They have their machinery with which they replicate, and they have the ability and capacity to transmit certain characters that are hitherto not in the original or receiving bacterium. We will consider plasmids as a topic in the course of this course. And so if you can see that from cell A, the chromosomal DNA is as you are seeing. On the right side of that cell, you will see the plasmid. So it has, by way of transformation, been sent into a cell, and you can see it incorporated in the second cell, as you have seen. So the exogenous plasmid is now taken over or taken in by a bacteria cell by transformation. It will no more not be the original bacterium with only its chromosomal DNA. Something is incorporated. So its activity, why it will transmit, the way it will exhibit, the disease it may transmit will be different from the initial or original or the donor bacterium, courtesy of the transformation process. Modes of gene transfer. Number 2, transduction. It is a process of genetic recombination in bacteria in which a bacteriophage are bacteria infected with viruses. So these viral particles or bacterium will now transmit a piece of this DNA from one bacterium to another. The bacteriophage will incorporate it into the bacterium that initially does not have and cause it to produce it. It can occur through any of these two processes, either by lytic process or by lysogenic cycle of the bacteriophage. Under lytic, following attachment and entry and replication and assembly of the gene that was sent in by the bacteriophage into the chromosomal DNA of the receiving organism or bacterium, the cell will eventually lyse, releasing the products containing the new DNA particles that were sent in by the bacteriophage. Under lysogeny, the process is almost the same, but the difference is such that the cell eventually does not burst to release the replicated DNA particle, but it will be there until the cell either dies, and eventually when it is now cut open, the incorporated particles are now released, and then the life cycle starts producing the other cells containing the incorporated particle. That is how it is run. Now, transduction has been found to occur in a variety of bacterial populations, including, but not limited to, 1. Escherichia coli, 2. Pseudomonas species, 3. Salmonella species, and Staphylococcus species, among other bacteria species. When you watch transduction on the diagram, you will see where the bacteria, which is chromosomal DNA, is having a bacteriophage attached to its surface. And you can see that it pierces open the bacterial wall and is inserting the viral particle or the gene of interest, such that at the second cell, you will see there is a recombination of the viral particle so introduced by the bacteriophage. Several copies of it were produced, and there will be recombination, and then daughter cells containing the introduced viral particles alongside the DNA fragments are produced. And when such cells are now made to undergo another cycle of transduction or reproduction, you will see that they will produce bacterial cells that will incorporate or will contain the particles that were introduced by the bacteriophage, as seen on the last cell, where the DNA, chromosomal DNA of the bacterial cell by homologous recombination is incorporated with what the viral particles, the virus, the bacteriophage introduced into the cell. Conjugation as the last of the three modes of gene transfer. In conjugation, DNA is transferred from one bacterium to the other through a tube from two physically contact bacterial cells. Get the difference? In this case of conjugation, the transfer is made between two physically contact bacterial cells. This is different from the other two methods. In the summary, I will bring out those differences in the three processes. Now, the donor cell pulls itself close to the recipient bacterium, using a structure called a pilus or pili in plural. DNA is then transferred between the two cells, and in most cases, this DNA is in the form of a plasmid. We've mentioned plasmid before, extra chromosomal DNA component that are capable of self-replication outside of the chromosomal DNA. The donor cells has a fertility factor called an F factor, which codes for the protein that make up the sex pili, which is that F pili. Now, if any cell that hitherto did not have the F factor, upon receiving the F factor, turns into an F positive donor and can also be able to produce its own pili or pilus in singular, and can be able to transfer DNA to other cells. Remember that we said earlier that the other receiving bacterium is pulled to the one having by use of the fertility factor, the F factor or the sex factor, which forms that tube or segment through which the gene of interest is transferred through that tubing from the F positive donor into the cell that was not having it in the initial stage, and upon receiving, it too now becomes F positive, so that it can also be able to transmit such a factor into other cells. If you consider the diagram, you will see chromosomal DNA and the F plasmid in the cell 1, which is an F positive cell as a donor, which is pilus, pulling a cell that was F negative because it doesn't have the sex cell or the fertility factor. So when they now come together and join, that tubing is made as seen in the second cells lying side by side. And you can see that one is only the one which is the donor that has the F plasmid. The second one does not. So through the tubing as seen in structure 3 or the cells in level 3, you will see that the pilus, the F plasmid now is being taken into the cell that did not have it initially, and eventually it goes, the septum, the tubing that was made in between through which the transmission was made, we now cut open, producing two daughter cells, now both of them having the F positive or the fertility factor, so that each of them independently will now continue to donate the sex pili or the transmitted gene. That is how conjugation takes place. Now the three processes viewed together in a glance is as seen. You can see under transformation the bacterium from the left. If you can see the red color, there has a gene that is or was not present in the B, which is the receiving bacterial cell now. So upon release of this DNA from the cell having it, there will be entry by way of attachment, by way of recombination and whatever, into the cell that did not have. And you know, some of these factors, as explained earlier, could be antibiotic resistant gene. Any of the such genes can be transmitted, such that the cell that ab initio was not resistant to any antibiotic may be made to be resistant to a particular antibiotic, courtesy of this type of recombination, donation from the one that has to a one that did not ab initio has the resistant gene, and it will continue rejecting or preventing the effect of such antibiotic in such an organism when it affects another person. Now the B, which has to do with the bacterial transduction, you can see the cell on the left being infected with a phage, and the one on the right initially did not have, but because it has let loose the phage, as I said earlier, either by lysogeny or by lytic cycle, it now introduces the phage into the bacterium or the genetic material with the help of the phage into the bacterium that did not have it, and upon receiving it, it will not be incorporated into the chromosomal DNA of the recipient following replication. And then in the last one, which is the conjugation, you can see that the cells comes together, form a septum, and the genetic material which is seen on the cell on the left passes through the tubing into the second cell that now contains the transmitted or transferred genetic material, which will be incorporated, and then the second cell will now be seen also to have that DNA component or the gene of interest. In summary, we were made to understand that in transformation, a bacterium is made to take up a piece of DNA floating in its environment. This can happen by use of, say, some specialized mechanisms of this thing called transposomes or jumping genes. They are suspended in the environment into which the bacterium that initially did not have this factor, these genes, is suspended, so they can jump into and be incorporated into such bacterium, and they also will have such a factor or such a character upon reproduction. In transduction, there is still transfer, and in this case accidental. But what is peculiar and key in transduction is that a bacteriophage, that is bacteria say infected by bacteria, is the factor or the vector which transmits the gene of interest. It is incorporated in the bacteriophage, and it now transmits onto the recipient, which now harbors this gene upon recombination. In conjugation, DNA is transferred between bacteria through two tubes, through two cells, by cells lying side by side through a tubing, by homologous recombination. And so you will understand that it is important we understand that these steps, some are natural, some in the environment, depending on whatever that we want to do. It may be that it is for therapy, and then it will be introduced. A special organism or gene will be incorporated and then introduced for the purpose of cure or therapy of any factor that is lacking in a patient. And so this gene transfer, in as much as it's transmitting diseases, is also of use. So depending on the context in question, it has advantages and there are also disadvantages. At this juncture, I want to thank you all and expect that as you read through during subsequent classes, you'll be made to ask questions, or you can send your questions online so that we can interact as the case may be, or during physical classes we can then meet to resolve issues that you do not understand, because the class is large and that is the essence of this visual presentation. So during physical contacts you can ask your questions, or if you can get yourself control, you can access online. If time could permit, we can interact online to save time and to gain more knowledge. I learn from you, you learn from me as we contribute. God bless you. Thank you.